When you listen to a story, like when your friend tells you about their crazy weekend or your mom would read you Harry Potter as a kid, how does your brain integrate the auditory information it receives into a coherent narrative? Do some brain areas respond best to hearing syllables, words, sentences, paragraphs, or entire stories? Communication through language is such a fundamental and unique part of the human experience, but how do we actually process all the stories we hear? Lerner, Honey, Silbert, and Hassan were wondering the same thing, so they ran a study which they describe in this paper called Topographic Mapping of a Hierarchy of Temporal Receptive Windows Using a Narrated Story. We know that space and time are two basic properties of the physical world. We already understand a lot about how information is integrated across space in our visual systems. Neurons in visual cortex have larger receptive fields as you move from early visual areas to higher level areas. For example, we already learned that primary visual cortex has cells that respond selectively to lines at particular orientations, while the higher level lateral occipital complex responds selectively to entire objects. This happens because the neurons in higher level areas integrate information from multiple neurons in earlier areas. Despite our understanding of how neurons integrate information across space, much less is known about how neurons integrate information across time. The authors defined the concept of a temporal receptive window, which is the time-related equivalent of a spatial receptive field. It's the length of time before a neuronal response during which sensory information can affect that response. The authors found that as you move from low-level to high-level visual areas, the temporal receptive windows get larger, just like what we already know about spatial receptive fields. This prompted them to ask whether their findings extend to other sensory modalities, so they tested how temporal receptive windows are organized throughout the auditory system. They hypothesized that, like in the visual system, temporal receptive windows increase gradually from early auditory areas to high-order language areas, with frontal areas being able to accumulate information over the longest window of time. The human auditory system is specialized to process language. Units of language, such as words, sentences, and paragraphs, correspond naturally to different timescales of sensory input, from short to long time periods. To assess the timescale of processing in the auditory cortex and association areas, the authors measured the reliability of neural responses evoked by different scrambled versions of a real-life 7-minute story. By comparing neural responses to these different scrambled stimuli, the authors wanted to find out how sensitive each brain area was to information that arrived at different points in the past.
This study had 15 participants aged 20 to 36 years old. There were 5 conditions, and data was collected from each participant until there was data from 11 subjects for each condition. A 3 Tesla MRI scanner was used for this study. Functional information was collected in 300 volumes using a T2 star-weighted echoplanar imaging pulse sequence. Each volume was comprised of 25 slices, which were 3 mm thick with a 1 mm gap, resulting in a plane resolution of 3 mm squared. The slice acquisition order for this study was interleaved. Anatomical information was collected with 160 T1 weighted high-resolution anatomical images. The voxels were 1 mm cubed, and slices were in the same orientation as EPI. Anatomical images were used for cortical recognition and 3D reconstruction. Stimuli for this experiment was generated from a 7-minute real-life story titled Pie Man by Jim O'Grady. Subjects listened to 5 different variations of the story. One was the whole story beginning to end. Another was the whole story waveform reversed in time. For the final 3, the story was segmented and scrambled into 3 timescales. To localize areas that reliably responded to natural stories, an independent 15-minute real-life story was played to each subject, and an inter-subject correlation analysis was used to create a reliability map that defined independent regions of interest. Pre-processing was used to reconstruct and analyze fMRI data. Spatial smoothing was applied using a Gaussian filter of 6 mm full width at half maximum value. Cortical surface was reconstructed from the anatomical images into 3D telluric space. Inter-subject correlation analysis, INTER-SC, produced the central results of this study. This analysis provided a measure of the reliability of the response to temporally complex stimuli, such as a story, by comparing the bold response time courses across different subjects. Therefore, a subject's brain response to naturalistic stimuli is used to predict responses in other subjects. The inter-SC map obtained for the main story overlapped with the inter-SC map obtained for the localizer story. The experimenters defined a set of ROIs by manually separating significantly responsive voxels in the independent localizer map into 5 adjacent subregions. 2 additional ROIs in the precuneus and the medial prefrontal cortex were defined from the localizer experiment. The experimenters also sampled all voxels along the temporal parietal axis, based on information from the localizer experiment, and calculated a temporal receptive window, or TRW index. Standard deviation was assessed independently for each ROI and each condition within each subject, and averaged across all subjects, and compared to determine the response amplitude. To unscramble the responses to scrambled stimuli, first the bold response for each segment was separated, taking into account the hemodynamic response function delay. Second, the natural responses were reordered to match the temporal order of the forward condition. Finally, the unscrambled sentence and paragraph response time courses were correlated with the response to the forward story. The backward story was also compared to the forward by flipping the average response in the backward condition in each voxel and shifting by 5 seconds to correct for hemodynamic delay. Early auditory areas such as A1 responded reliably to all conditions regardless of their temporal structure and exhibited short TRWs. Rostrally and caudally from A1 to higher level areas, the reliability of responses became increasingly dependent on the level of stimulus-preserved temporal structure. Areas adjacent to A1 along the superior temporal gyrus exhibited intermediate TRWs, and the longest TRWs were found in the posterior superior temporal cortex, the temporal parietal junction, the precuneus, and the frontal cortex. In the later regions, the reliable responses were evoked only by paragraph scrambled and intact story conditions. A hierarchy of timescale sensitivity was also observed in ROI analysis. Early auditory areas, such as A1, exhibited high correlations for all conditions, suggesting that the reliability of responses in this area are not dependent on coherent temporally extended structure. Moving rostrally from A1 toward the temporal parietal junction, the reliability of responses becomes more dependent on the intact temporal structure. RO2 responds to words are longer, RO3 responds to sentences are longer, RO4 responds to paragraphs are longer, and RO5 responds most reliably to the entire story.
The TRW hypothesis predicts that a region with a given TRW length will respond similarly to a coherent event of that length regardless of the larger temporal context. To test this prediction, the time course observed from the intact story was compared with the unscrambled versions of time courses observed from both the sentence scrambled and paragraph scrambled conditions. After unscrambling the brain responses, feedback from A1 Plus was similar across all conditions, regardless of the temporal order. Also, the TRW within the temporoparietal junction coincided with the timescale of the entire paragraph, as the responses to the intact story were only correlated with the unscrambled paragraph's time course. Strong correlations were observed when comparing the intact forward story with the time-reversed back story, the unscrambled sentences, and the unscrambled paragraphs. The similarities were observed in the raw unscrambled time courses as well as in the mean inter-SC coefficients. Thus consistent with the hypothesis that A1 Plus has a short TRW, responses in A1 Plus were found to be induced by moment-to-moment auditory input separate from the temporal history. The TRW within the posterior superior temporal gyrus was found to coincide with the timescale of a single sentence, after observing that the time course of unscrambled sentences correlated with the responses to the intact story.
The longest TRWs were observed in the precuneus and medial prefrontal cortex, shown here. The precuneus responded reliably to both the paragraph scrambled and the forward story conditions, and its responses were similar for the intact story and for the unscrambled response to the paragraph scrambled condition, which showed that processing in that area occurs over long timescales of around 30 seconds. The medial prefrontal cortex only responded reliably to the forward story condition, and unscrambling analysis showed that its activation time course during the intact story did not correlate with unscrambled patterns from any of the other conditions, so it seems to process information over long timescales of entire stories. Interestingly, disrupting temporal order had no effect on bold response amplitudes, even in brain areas in which it dramatically reduced response reliability. This figure shows that the standard deviation, or dynamic range, of the bold responses within each brain region was similar for all scrambling levels. This shows that there's a clear dissociation between response reliability and response amplitude of the bold signal, so measuring response reliability can uncover effects that response amplitudes don't reveal.
So how exactly do these results fit with the wider literature in the area? The authors relate several findings to numerous studies, a factor which further establishes their authenticity. Such is the case of brain regions providing context for one another's processing, first proposed by McIntosh in 2000. However, back then the idea wasn't investigated beyond short semantic words and sentences, while in this study the researchers were able to identify 3 distinct processing stages from the incoming audio soundtrack. First, perceptual sensory areas analyze low-level acoustic properties through rapid processing and then encode the low-level information from this, regardless of its content or meaning. Secondly, high-order linguistic areas process lexical items and grammatical structures within the boundaries of a single sentence. This corroborates a similar finding by Okada et al. That core auditory regions are particularly sensitive to acoustic features, whilst downstream regions show greater sensitivity to speech intelligibility. Thirdly, extralinguistic areas in the cortex were observed in the precuneus, the medial frontal cortex, the inferior frontal gyrus, and the paracingulate cortices, all of which coincide with similar observations made by Scott et al. On levels of speech comprehension in these brain regions. So what do researchers hope to accomplish with these findings? Well, one of their biggest aspirations is to link the notion of TRWs with existing bodies of research on working memory and predictive coding, both of which are essential for semantic and syntactic processing in language and communication. Basing their findings off of the Keeble et al. 2008 prediction of working memory, it is suggested that regions with longer TRWs will make predictions about forthcoming events with correspondingly larger timescales. This promises to be a new and exciting development in the wider literature, and is one of several areas that warrants further research to fully investigate the relationship between the timescales over which neural circuits integrate information from the past and the timescales over which they forecast the future. Future research will also be necessary to explore how information is accumulated over long timescales and across modalities in the frontal cortex. Combining the TRW map obtained using the scrambled sequences of a narrated story with the TRW map of those in a silent movie indicates that longer TRWs seem to be multimodal, as they clearly process long temporal structures whether through auditory or visual means. The maps mostly tend to overlap in high-order areas such as Brodmann 39 and Brodmann 40, both of which are known to be involved in semantic processing. There are several limitations that bear mentioning in this study. The authors failed to mention how many of their participants were male or female. As clearly demonstrated by Bell et al. In 2006, males and females exhibit differential performance in various cognitive tasks, including verbal ones, so gender may have had an effect in this study even though this information is not provided. 15 participants took part in this study, however data was only collected from 11 subjects for each of the 5 conditions. Extensive scanner time for participants led to confounds in the data such as motion artifacts, which along with signal corruption was responsible for data not being collected from all participants across all conditions. Previous research by Pajula and Toka in 2015 has found that 20 subjects is the minimum number needed to obtain highly reliable inter-subject correlations. This study's sample size of 11 thus raises concerns on the generality of the TRW findings. These implications may or may not be enough to confound the data, but bear mentioning given the failure of the authors to do so.